Welcome to our Fab Night Out. It looks like all of our presenters are in, so we will get started. It's a jam-packed hour. My name is Catherine Febria, and I'm the director of the Healthy Headwaters Lab and faculty member at the University of Windsor. And so as we gather tonight, most of us from our homes or some of our advisory board members on a ferry to Pelee Island, I wanted to thank you all for choosing to spend this next hour with us. And as you know, we're here from different pockets of Essex County and Windsor and Southwest Ontario, and just wanted to acknowledge that we're on the traditional territory of the Three Fires Confederacy of First Nations, the Odawa, the Ojibwe, and the Potawatomi. And it's with that sense of gratitude that our work and our team are able to do the work that we do, and working with stewards of the land is kind of our thing. And so today is a very exciting night for us. We've hoping originally to do this in person, maybe at GLIER or somewhere in the university where we could bring people together. And so that's the intention for this next hour, is to talk to you about the research that our team is doing, but also all the partnerships that we're trying to align and maximize in the name of freshwater and farming sustainability. So with no further ado, I will just walk you through kind of our plan for tonight, and we'll take the next hour sharing the floor with different people who are wanting to share their stories or their narratives and the ways that we're all hoping we can work together. So if you come out of today getting a sense of what our lab is doing, but also what partners are doing, and maybe find ways to connect with other researchers, practitioners, or even individual farming partners in this circle, then that will be a success for us. So just a couple of housekeeping things. On the technical side, you should have received information from Jess on our team. The presenters have already, you'll be getting the hosting permissions as I introduce you, or as Jess introduces you. And we hope we can end this hour virtually connecting in breakout rooms. So the program for today is, again, introducing myself. You'll hear from various members of our team. And we also really wanted to take this night to celebrate our advisory board members and their kind of commitment to engaging in this space with us. We'll go through some research updates, and then we'll go through some really quick lightning talks. So 5 minutes or less, a few slides, no more, and different researchers and partners will tell you a bit about the work that they're doing here in the context of Essex County and Southwest Ontario, and hopefully open the floor to more connections and within the community and with one another by the end of this evening. So if you don't have a cup of tea or a glass of wine or something to nip popcorn to snack on, just enjoy this next hour. Okay, so I wanted to acknowledge our advisory board members who are here tonight. Mostly, I think they're all here virtually, which is wonderful. And so this is a process, thanks to support from the Faculty of Science at the University of Windsor in our Canada Research Chair position. We've been able to really invest in social infrastructure to support the research in our lab. And so I wanted to thank these environmental leaders: Henry Donater, Jesse Dreger, Summer Garcia, Clint Jacobs, Dean Martin, Chad Quinlan, Justine Taylor, as our first inaugural advisory board members. Most of us have met before the pandemic, just when we launched the lab, to talk about, how do we even do something like an advisory board and center farming communities in a water research lab. And so we landed on this format. And it allowed us to not only amplify the work and sort of really get to know the farming community here in Southwest Ontario, but then equally provide a way for farmers to tell us and share with us the kinds of questions that they're grappling with at the farm field, and how that links to conservation and stewardship of this area that we call home. And so on this list are farmers that are multi-generational farmers here in Essex County. Two are from Bequejenon Territory, Walpole Island First Nation, including one that's new to farming in the effort to rematriate native corn back to Walpole after generations of being not grown in that territory. We also have our partner at the Ontario Greenhouse Vegetable Growers Association, Justine Taylor, and we have multiple farmers here who have been, welcoming our team throughout the pandemic to sample soils and really test some of our systems in terms of trying to do science in a really effective, safe, and impactful way. So thank you to our advisory board members. We'll talk business at the end of this hour. So next, For those of you who are just learning about the work that our team does, I'll just give you a really brief synopsis of the work. Ultimately, we're interested in restoration in agricultural landscapes. So finding and advancing research that promotes freshwater and farming sustainability. This pictograph in the middle sort of just captures some of the science we're talking about today. And the dots in yellow to your right are the talks we'll just be giving a nod to in different ways. But ultimately, we're trying to understand how the farming practices here in Essex County and Southwest Ontario on clay plain soils and the different practices, whether it be cover cropping, organic farming, various tillage regimes, the presence of wetlands, how that ultimately influences the nature of organic matter or carbon in soils. And understanding that will help understand many other research partnerships who are looking at phosphorus, for example, runoff via tile drains, looking at the drainage system in terms of its flood capacity and resilience for flooding and climate-related changes, and runoff to the Great Lakes that we all care about. This is all leading to rethinking, how can carbon be a potential tracer of our practices on farm into the drain network, and also rethinking around drains themselves. Can we think about what's being planted and how they're being managed to rebuild multifunctional systems to allow things like pest control or pollination, and thinking about building the baseline in this next year or two to inform stacked BMP implementation, and thinking about the role of social dimensions in how ecologically minded or ecologically motivated actions on a farm can be implemented and accelerated through different ways of knowing, whether it be traditional ecological knowledge or social learning between farmers and that sort of thing. So it really is laying the groundwork for our social ecological experiments in the real world. Ultimately, we think this can directly apply to just questions around species at risk, which we know are present in many of these farm landscapes. And ultimately, we also know that some of the solutions will lie in the effectiveness of nature-based solutions, whether they be wetlands in combination with drains, whether it be tools for Phragmites in combination with a tillage practice. So looking at how can we use our natural landscapes in our communities to help create gradients of experiments and combinations that we can all address together. So we're just a freshwater ecosystem lab. We can't do it all, which is why we invite partnerships in this. Thank you, Jess.
Can you see my screen there?
Yes, that's great. Thank you.
Okay, let me just get set up. So hi, everyone. As Catherine mentioned, my name is Lauren Weller. I'm an MSc student in the Healthy Headwaters Lab, and I'm working on carbon complexity in agricultural landscapes. So why do we look at carbon complexity? Well, we know that globally we're facing a multitude of challenges with both freshwater quality and soil degradation. Conservation agriculture, specifically investments in soil health, have been touted as the solution to both of these problems, but their effectiveness has been really hard to quantify. And this is because studies have traditionally been restricted to either freshwater or soil science with few multidisciplinary investigations at the soil-water interface, particularly in agricultural landscapes. So in soils, carbon, specifically soil organic carbon, is often used as a proxy for organic matter as it's more easily measured. Soil organic carbon is considered a reliable indicator of soil health, which is not surprising because it's fundamental to many processes, particularly nutrient cycling. And organic matter plays a similar role in freshwater ecosystems. In this way, organic matter, specifically dissolved organic matter, acts as a bridge between the land and the water, with much of DOM entering the freshwater either within soils or being transported via soils to the freshwater environment. And you can have a look at this little diagram here that really kind of lays it out. So we end up with like root litter, which is input, and then all these processes are happening within soil, with eventually dissolved organic matter being exported into the freshwater environment. So what is DOM and how can fluorescent DOM help us to understand it? What we commonly refer to as dissolved organic matter is in fact hundreds of mostly uncharacterized complex compounds, varying from simple molecules through to highly complicated aromatic molecules. Recently, we've come to understand that these substances are in fact recognizable compounds but undergoing decomposition. So picture, if you will, a pile of sweaters, each being unraveled, but at different stages. Some have arms, maybe some just have the body, and some just have the collar left. So analysis can show us the wool blend, in this case, the amount of carbon or nitrogen, but establishing what we're actually looking at in terms of an item of clothing is sometimes a challenge. So fortunately, DOM has certain fluorescent properties, which allow us to study and understand the underlying complexity. So light at a certain wavelength is passed through a liquid sample to create what we call an EAM. And different substances fluoresce at different wavelengths, so groups of molecules can be identified and thus characterized. Which brings me to the goal of my research. As the title of the presentation suggests, I'm seeking to understand carbon complexity in agricultural landscapes. We know we can use fluorescent DOM to distinguish between broad land uses such as urban, forest, and agricultural sites. But can we also use fluorescent DOM to confer specific field scale management processes? I'm of course hoping that we can, and I hypothesize that we will see specific fluorescent DOM signatures emerging along a gradient of management practices. So to this end, in order to test my hypothesis, we established a gradient of management practices currently being undertaken in Essex County. And these include modern conventional agriculture with regular tillage, a no or conservation tillage regime, a no or conservation tillage regime with cover crops. And then we also included a forest woodlot, a forest and woodlot site to act as a reference for natural soil conditions. My research was place-based here in Essex County. As Brookston clay is the dominant soil type for this region, it's shown here in red. We decided to stick all of our sites on Brookston clay to ensure that any fluorescent DOM signatures that we managed to obtain would actually reflect the management practices rather than the inherent soil qualities. Agricultural fields in Essex County are also largely tile drained, and so they offer us a unique opportunity to study how soil management impacts freshwater systems, as both the soil and the tiles can be surveyed individually to understand DOM and nutrient dynamics associated with a specific individual field. Furthermore, we know that agricultural areas are associated with a decrease in structural complexity of DOM, which is exacerbated by tiles and channelized drains. Thus, tile drained landscapes remain key in tackling the joint challenges of soil and freshwater degradation. By sampling both the agricultural soils and the water entering the freshwater ecosystems via tile drains, we hope to better understand carbon complexity in both systems as a step towards understanding the role of soil health in freshwater restoration. So we managed to get some field work done over the fall, despite the challenges brought to us by COVID. We managed to complete our fall soil sampling survey. At each site, 10 boreholes were dug. It worked out to 1 per acre in a 10-acre plot, and each borehole was dug down to 60 centimeters using a soil sampling auger. Soils were then collected in buckets separated by depth, with each bucket representing a 15-centimeter increment, and these were mixed before being subsampled, and then the boreholes were rehabilitated. At each borehole, a measurement was also taken with a soil and moisture probe, both at the surface and at depth, and a water infiltration test was completed. We also dug a small, shallow test pit in order to assess visual indicators of soil health. Following the field season, all samples were frozen, and this is where I am now, although this freezer is considerably more full, I will add. So my plan for the rest of the winter is to process the rest of my soil samples through dehydration, crushing, and leaching. Moving into early next year, we're hoping to run all of our samples through the Hariba Aqualog, which will give us that fluorescent DOM signature for each sample. We're then hoping to build something called a PARFAC model, which will basically help us to categorize each of these samples into groups, so we'll be able to see distinct signatures, hopefully, for each land management type. We're also going to be analyzing these samples for soil organic carbon as a test back to data that farmers routinely collect, which is soil organic carbon, nutrients, and as well as nutrients. So we can sort of understand if there's any link there between the EAMs that we're seeing and nutrient dynamics, both in the tiles and the soil. And then moving on to the spring, we're hoping that if all things go well and COVID does not shut us down again, I'll be able to do a tile grab sampling campaign that'll coincide to time with the spring melts. And then moving on into the summer and fall, we'll be doing data analysis and write-up. So thank you very much. There's a lovely picture of us working out in the summer during our field season.
Thank you, Lauren.
It's okay.
I'll now switch back and take the screen over. Can you come back? Are we now? I'm going to just make sure I'm sharing my screen. Can you see the our one screen? Are we back to our home page? Cool. Okay. This is the lovely thing. So thank you, Lauren. So as you can see, Lauren's work covers just sort of that critical baseline to understand what's happening at the farm level. I wanted to thank, we have multiple MOUs with individual farmers from across the region that enabled Lauren to be able to do that work. And so as we move into the winter and the spring, each of those individual farmers will actually receive scans of their soils and answer a critical question about whether or not those farm practices are making a difference, and allow us, as we continue to expand our baseline over larger gradients, a way for us to have something to talk about with the farmers and see how well and how responsive something like tillage over 20 years, how does that compare with something that was maybe no tillage regime of 3? How, once we start expanding next into the other types of soils, you know, how consistent are those emission excitation matrices? And I love that wool sweater analogy, Lauren. Thank you for that. So, you know, critical. So as we talked about Lauren's work focusing very much at the farm field, we have research from undergraduate students like Cheyenne Nolan who are working in the drain space using a standard test for measuring carbon processing called carbon processing in these drains. She's looking at both urban and agricultural drains here in Essex County and using this organic matter signature and processing in our stream systems to understand whether or not agricultural drains and urban drains operate similarly or differently. So she has cotton strips at the moment deployed around the region, capturing some critical baseline information about energy sources in Essex County waterways. Emily Brown is also continuing to expand this work into looking at best management practices. And in partnership with Nature Conservancy Canada and their Marion Gerlain Nature Reserve property, we're implementing the baseline ecological monitoring under soils and sediments with and without frag control, and in farms that will serve as a baseline to future stacked BMP trials. So we'll be able to generate information that expands upon what Lauren has just described at the farm field to those that have best management practices already in place, and look and add the dimension of what happens with and without frag management, and what is the characterization of existing wetlands that are 10 or so years old in this landscape. I'm going to go to my next slide, which is not cooperating at the moment, but bear with me. And I'm sorry, it's thinking. And I'll move to our next slide, which is our project in the riparian space. And so while that's loading, I will also mention that the other graduate student that's then, as we move from farm field, here we go, it's coming. Right, so as we move from farm field, which is what Lauren talked about, into the drain space, which is what Cheyenne will be working on, and thinking about other BMPs, we also have work that's happening in the drains themselves. And again, through partnerships that we've been able to initiate over this past year, we'd like to acknowledge those partners. And that includes, again, Nature Conservancy Canada, but also through partnerships with the Kingsville, Ontario's Drain Superintendent, ERCA, and St. Clair Conservation Authority. We're starting to think about vegetation and the nature of these drains. Can we start to build and use biodiversity in a functional sense, so that we're starting to think about whether or not there are certain plants, certain species that we could use that might offer pest management functions, drainage, flooding mitigation functions. And so I wanted to acknowledge our partners in a project led by Ryan Graham and our team. Okay, so finally, we will move to this next component, which is really about the partnership network. That's the point of today. We really see this as being vital to the work that we're doing in the restoration space. And so for that, I wanted to switch to our lightning talks. We have here tonight We have two different speakers from ERCA, some speakers from the University of Windsor, and Nature Conservancy today. So this is all driven by a larger vision. Some of you might know that we are on the doorstep to the decade of restoration. And if you look at this effort, which is around that many countries around the world have really embraced, it is an effort that over the next decade to focus on soil conservation. So we know that the solutions are really based on the land. And a lot of the efforts around this next decade is around trying to find ways to finance these restoration actions, whether they be stack BMPs, empower partnerships, like really celebrate our leaders who we have here tonight, really think about research that can think about how we can change behaviors, change the culture, invest in research to do all of these things. And specifically here in Canada, if we can do all of those things and really give the floor and make way for Indigenous self-determination, this is an act of reconciliation. So for tonight, I just hope that you'll help join me and connect with one another to really think about how we can accelerate partnerships here in Essex County, how research communities, we're here to listen and learn from our partners. And hopefully this is the start of an annual tradition that we have to come together and give people updates and continue to further the work that we know needs to happen here. And if we can be as inclusive and diverse as possible from the start, I feel like we're in a good space. So part of that is our turn to listen. So we will give the floor now to our speakers. And I will invite our first speaker, our parent speakers are from ERCA. And so we will give the floor to them and again, enjoy the talks.
I guess that's me. I'm just going to share my screen. Thank you so much, Dr. Febria. Let's see. There, how are we doing? Can you see?
All good. Perfect.
Great. Okay. So I'm going to jump right in, 5 minutes, no more. In 2018, in response to severe flooding, significant shoreline erosion, infrastructure failure, new and renewed threats from invasive species, and a host of other issues, ERCA initiated a regional climate change strategy with municipalities and other partners, including the University of Windsor. This collaborative called the Windsor Essex Climate Change Collaborative includes a number of sector-based themes, including human health, built environment, natural hazards, agriculture, nature and ecosystems, and energy. And for each of these sectors, we really wanted to identify vulnerabilities and likely impacts resulting from climate change projections in order to inform action. So while some sectors are certainly easier than others, I'm not going to lie, agriculture is one where I'm struggling a little bit, and I'll tell you why further on. But really, climate change is of two pieces. There's the mitigation side, where we're still trying to prevent the worst impacts from occurring by reducing our greenhouse gas emissions. And on the other side, we have adaptation. So still we need to respond to the impacts of a changing climate, which we've already seen. And we've seen extensively throughout this region. So in terms of farming, I think some aspects of climate change look quite promising. So adaptation or climate change isn't necessarily all bad. There are opportunities with climate change as well, and including longer frost-free seasons, increases in growing degree days, and even increased atmospheric carbon dioxide can, in theory, lead to better crop yields and productivity. So that said, we are also experiencing an increase in the frequency of extreme events that can adversely affect all of these things. So the Ontario Federation of Agriculture believes that farmers are a solution to climate change. And as effective managers of the carbon and nitrogen cycles, farmers are in a really unique position to provide further greenhouse gas mitigation opportunities. We're currently working on a regional energy plan. That energy plan doesn't include land-based uses, but certainly I think this is an area where continued research is necessary to develop and expand the scope of these opportunities and develop both acceptable programs that will potentially incentivize adoption. And on the adaptation side, we know that impacts will be highly variable and unpredictable. And OFA recommends that the government should increase investment into adaptation strategies. The National Farmers Union has long seen an alignment between the farming crisis and the climate crisis. And now I might add that we now have the COVID crisis as well. So three crises coming together. And I think certainly the federal government is seeing COVID as an opportunity in terms of green recovery. And so, I think there's certainly opportunity. There are three main direct impacts of changing climate that will give rise to an uncountable number of indirect or secondary impacts. And the first is temperatures. So it's going to be warmer. You can see by this graph, we're looking at by 2080, upwards of 60 days over 30 degrees. So heat waves, droughts, fewer colder days, mean and minimum temperatures, daily temperatures projected to significantly increase in every season. It'll be wetter, so we've already seen this. Annual precipitation is expected to increase. Winter and spring will get wetter instead of snow, with a slight decline in the summer. And then, of course, wilder. So we're seeing more intense precipitation events that are expected to occur more frequently. On average, more rain is expected to fall faster during storm events. And of course, we've seen this throughout the season or throughout the last few years. Last year, our agronomist, like many other farmers in the region, couldn't plant corn at the demonstration farm. He had to plant soybeans. And we know that planting soybeans year after year is bad for soil health. Our forestry team also couldn't plant 20,000 trees planned for the region because they couldn't prepare fields in time. So, when I start looking at this and adaptation actions, I'm not going to lie, I know that farmers and when I have, informal conversation with friends about this, farmers are used to planning for uncertainty and are probably the most adaptive workers in any sector. So, farmers are adapting on a daily, weekly, and seasonal basis according to the weather. So, it certainly makes me think, where can we take this work further? And I think it's important, like in any other sector that we're working through, I think it's important that we do put a climate lens over everything we're doing these days to try to understand and potentially get ahead of issues, and whether that's making the region more competitive for government funding or using it as using climate change as a driver for change. I'm not sure that it matters, but for what reason we use climate change in this sense, but I think that we can use it to certainly drive change. So just two more slides. We had some students look at agricultural climate change adaptation strategies from other regions. They looked at temperature and precipitation and impacts for grains and legumes, tomatoes and fruits for the region, and provided recommendations under these following adaptation actions. And again, it's not rocket science. I think we've been looking at these adaptation actions for years because we've been seeing the impacts for years. But with the projected climates, we know things are going to get worse. So where do we go from here? I think we continue to look at adaptation planning and consider the sector's role in mitigation by doing some of that modeling of potential carbon sequestration, impacts or their role in potentially bioenergy for the region, and then continuing to implement BMPs, support collaboration, and encourage and partner on research. I think there's a wide range of research opportunities, certainly for the region. We've already seen that from Dr. Febria's work, but working towards, things like rural food security, soil health, for increased resiliency, as Dr. Febria already mentioned, green infrastructure, and then, everything from renewable energy and generation on farms. So 5 minutes, I'm done, but here's my contact information. I'm curious, I'm interested to hear what is said in the breakout sessions, and I look forward to having further conversation.
Thank you, Claire. I'm going to ask, I wonder if you can share your screens with us after. I think for some of our screens, I think your Zoom screen covered some of the text. So we'll circulate maybe like a shortened bit as part of our follow-up with the survey that we're sending around to people. But thank you so much. We'll just be transitioning to Katie, giving her the floor to also from ERCA. So Dr. Katie Stamler is also an adjunct faculty at the University of Windsor at GLIER. And we'll just pivot to Katie when she's ready. And thank you again, Claire. You just have to unmute yourself, Katie, if you're not already.
Sorry, the controls disappeared on me when I started sharing my screen.
It always does that. Over to you.
Okay, awesome, cool. So you can see my screen, okay? Perfect. Okay, so I'm going to talk to you about about our regional phosphorus management plan, and it really goes together well with what Claire was just talking about. So we're trying at the Conservation Authority to really try to take these regional approaches where we know that phosphorus reduction and climate change, these aren't specific to certain areas. This is something we need to tackle as a region. It doesn't matter what watershed you're in, what municipality you're in, what sector you're in. So the reason that this came about is because of the harmful algal blooms in Lake Erie. You can see from this photo that there's a pretty persistent bloom as well that we get in Lake St. Clair. And this has gained a lot of international attention, and we know that we need to take action on it. And there's a lot of different factors that have resulted in a recurrence of the harmful algal blooms. And like Claire mentioned, climate change is a huge one for this. We know that it's warmer. We know that it's raining at a time of year when when the rainwater is carrying a lot more of the nutrients off of our fields and into our watercourses. We know the type of algae blooming are different and that they like the warmer temperatures that we have. We have a loss of wetlands, which are like nature's kidneys. So we know that we're not filtering the water the way that we should be when it's leaving the land. But the one thing that we're able to manage and that we're putting our focus and attention on is phosphorus. So we know that we have these targets of 40% reduction of phosphorus. And why that's important and why that came to be is that there's some cool science by David Schindler years and years ago that tells us that phosphorus is the limiting nutrient for algal growth. And we know that when we reduce phosphorus in the past, that it helped us to gain control over these harmful algal blooms. So that's what we're doing again. But before we were able to tackle those point sources, and now we have to go after non-point sources and try to reach that 40%, that further 40% reduction. And it's going to be hard, and we all know that it's going to be hard. So, how do we try to come up with ways that we can try to achieve that 40% reduction locally using solutions that actually work here? So the phosphorus management plan that we're working on it helps to achieve one of the goals of the Canada-Ontario Lake Erie Action Plan by supporting watershed and nearshore-based strategies and community-based planning for reducing phosphorus loading. So this is directly related to that action plan. And our goals for this are, we really just want to try to summarize what we already know about the Essex region. We want to try to identify where our knowledge gaps are so that we can try to direct research where we need research done so that we can have a better understanding of what we don't know. Trying to understand the sources and delivery and fate of phosphorus, determining those optimal phosphorus reduction strategies that work here locally, and making sure that we're aware and spreading that outreach and education, and that we have a clear strategy. So similar to how Claire has a working group, we have a similar structure where we have a steering committee made up of members from these different working groups. We have our research working group that includes a lot of members from the Healthy Watersheds Lab, our municipal working group that we're still putting together, and that agriculture working group that I'm hoping this Farm Advisory Board will choose to support and will represent and be on this agriculture working group to help us make sure that our local farmers are represented and that the challenges that they face are included. So I'm just going to run through super quick what we already do know. So what we know is that Essex is unique. If I had $1 for every time I said Essex is different, I would be a rich lady. So we know that the Essex region has several small watersheds that are completely independent. They cross municipal borders, and we have 3 big watersheds really that are draining to our 3 major water bodies. As Lauren said, we know that we're dominated by Brookston clay soil, which comes with its own challenges for certain BMPs that other people can use with ease. We have very flat topography, which has its own challenges. And we have a lot of agriculture, right? So our land use in the Essex region, we've got a few urban centers, We've got mostly row crop agriculture, and then we have the unique challenge of having a lot of greenhouse agriculture around Leamington and Kingsville. We know we have very bad water quality in our tributaries. We do the provincial report cards, we score on average a D-. The provincial average is a C. So we know that we have some work to do here. I'm just going to highlight a couple differences between our greenhouse influence streams and our agriculture influence streams. And we do know that phosphorus concentrations are higher from our greenhouse streams. We also know that the timing is different. So we have higher phosphorus concentrations in the summertime in those greenhouse influence streams. And in our agriculture, row crop agriculture dominated streams, our highest phosphorus happens in the winter during events when you're getting that runoff when there's nothing planted on the fields. So we also know a bunch of solutions. We know that our greenhouse sector is working very hard on a lot of strategies to minimize their inputs, maximize their reuse, and manage their excess. And they have lots of collaborations, and they're making good progress on this, and they are a very willing participant in finding solutions to this. We also know there's tons of agriculture strategies. The two key ones here are the 4 R's, the right source, rate, time, and place. And so much can go into the 4 R's. This can also incorporate your tillage practices and all of those sorts of things. So making sure we're using fertilizer correctly. And when I talk to the public about this, I always say that it's our responsibility as homeowners as well to make sure that we're following the 4 R's and only using any product like fertilizer as we need it. And planting cover crops is a great agricultural practice that we know is challenging in the Essex region because of those clay soils. So finding those optimal cover crops. This is just a good visual to show you on a field that Henry planted for us, where he planted cover crop on one side and left the other side bare with just wheat stubble. And you can see how much more soil came off his field where he had no cover crop. We know there's great drainage solutions for reducing erosion. Our municipalities are working hard on their bigger infrastructure solutions for reducing CSOs and putting in retention basins. And we have lots of individual and community strategies as well. Our tree plantings, making sure that we're keeping things out of our waterways that don't belong there, maintaining our septic systems, and if we're able to, planting rain gardens. So that's just what I wanted to talk to you about today. So that's what we're working on. And trying to figure out again how to make that the best, most local solution to phosphorus management here. So again, there's my contact information. I think it'll be available later on as well. Thanks for the opportunity.
Thank you so much, Katie. And so aspects of her talk we'll bring back to the FAB in our business meeting shortly, but there's plenty of room for those of us who are here today who are wanting to engage. So we have a breakout room specifically to meet with both Claire and Katie. So we're now going to shift to a series of research talks. And so I'll just invite Dr. Cameron Proctor, a new faculty member in the School of Environment, to share the floor with us. And then after that, we'll go to Mike McKay and Ken Drouillard, and then we'll end with a Nature Conservancy lightning talk. So thank you very much, Katie, and Cameron, over to you.
Okay. All right. Thank you very much, Catherine, for inviting me here. I'm just going to see if I can share my screen here. Okay. So probably what's going to be different probably from the talks so far with my talk is this is a lot more narrow. So what I really want to talk about is some of my bigger research interests, which is looking at plant microbe interactions in soil and the carbon that comes off roots. So one of the things that's really kind of important about roots, I probably don't have to talk to you guys too much about this, is a very root heavy group, which I'm not used to talking in. But what I really want to do with my research is to design better roots. And to do that, we need to know a lot more about how they operate, their biomass, their activity. And so far, activities and research below ground has been kind of hindered because they're not really as readily apparent. But in my world, the boundary between soil and roots is one of the most important boundaries in the entire world. It controls how much nutrients go into our food web. It controls overall biomass. So what actually happens in those few centimeters is really hard to get at, but it's very important for reducing fertilizer use, improving yields, and overall promoting soil health. Roots kind of cause this sort of carbon cascade, which plant photosynthase go to leaves, go to roots, go to plant microbes, go to soil. So all along that particular chain, I'm interested in all of those particular interactions. And we also know that roots are very important. I mean, up to half of all photosynthase are going to be deposited below ground. There's a reason that we call roots the hidden half, because a good chunk of your plant is actually below ground. And a good part of that is biomass that's going to be sort of left over year. And a good part of that is just sort of maintenance costs and roots sort of shedding compounds. And my big takeaway is that not all plant roots are the same. They differ in terms of their architecture, they differ in terms of their number of different root orders, and even an individual root can be extremely different from its neighbors. Some roots are active and they actually involved in the uptake and efflux processes, but some are just involved in transport. They're kind of like a highway system. So you have your in ramps and off ramps and stuff, and your bigger sort of larger railways, moving carbon up and moving carbon down and nutrients and water up. So a lot of my research is looking at what sort of architectural traits in the roots are best for things like drought resistance, promoting good plant microbial interactions. And I want to know basically how roots are going to adapt to things that we expect to see in the future, like a wetter, hotter environment that I think Katie and others mentioned. So one of the things that I tend to sort of narrowly focus on is this idea that plants can lose carbon through their roots, and that process is partially passive, but the roots don't have that much control over it. But plants can actively sometimes shed compounds. They do this to protect themselves from plant pathogens. They do this to promote beneficial systems. And these are extremely important processes. In fact, what we know is that if roots don't really have enough room to grow, if plants can't actually spend their carbon on growing roots, it actually reduces photosynthesis. So if you have the creation or the acquisition of carbon in leaves is actually just as important as how much they spend below ground. But we don't really know exactly a lot about that process, how that process changes over time, how it responds to stressors. And what my research is really trying to do is to try and figure out how do we have these above and below ground linkages, and how does this carbon, the composition, the magnitude of carbon, the quality and quantity, as well as its functional significance in many below ground important processes like uptake, et cetera. And also, we also do know that a lot of other environmental factors affect how roots put carbon below ground. If you grow plants too close to each other, root system doesn't grow, and they actually can even compete with each other, and that's considered to be sort of a loss. So it's really challenging to figure out these processes because some roots and some plants can respond cooperatively and some plants can respond competitively. And that's even more exaggerated when they bring the microbial community in, because sometimes plants work well with them as a symbiosis, and sometimes the soil microorganisms can attack them. So it's kind of a sort of a weird dance where you can't have too much being shed below ground, because you can actually attract pathogens. So a lot of my research is looking at the conditions that make plant roots more vulnerable to root pathogen attack. So going into two sort of things that I really want to do to sort of learn more about roots is sort of one, learn more about the biomass. We do a lot of work in plant phenotyping where we develop specific cultivars that are supposed to be drought resistant, and they might be drought resistant in the lab. When you bring them out into the field, they tend not to have those same properties. So actually monitoring what's actually happening in the below ground root system is really important. So we have these mycorrhizatrons that are basically clear tubes where you can put a camera down and watch how the plants grow. We also do this a little bit in the greenhouse as well, which gives us a little bit more control. So because the amount of biomass controls the interactions with the microbial community, we really want to know a lot about the properties of it, not just the total biomass, but the actual architecture, because how roots actually interact in the soil can actually have a huge implication for their nutrient uptake. And what we're also interested in doing is looking at root activity, the quality and quantity of carbon that's lost and its below ground function. So what are we losing below ground? Are there situations where we're losing too much carbon and it's sort of detrimental to the plant? Or are there situations where the plant and microbes are actually working pretty well in balance and we've minimized the amount of loss and maximized the amount of gain? And so understanding what carbon is present, is coming off these roots is extremely important. And just to sort of talk a little about some of the future projects that link into some of these things that I'm talking about. So what I really want to look at too is agricultural root response to drought. If we mimic climate change, if we reduce the amount of water coming in, how does the plant root system respond? Are there particular types of species that have particular types of root properties that actually respond really well to Essex drought? So not drought in other regions and other soil, but specifically to Essex County. We're looking at things like biosolids and roots. So I have a project right now where I'm looking at biosolid microplastics and its effects upon roots. Is it an interference agent? Is it hurting? Is there any sort of threshold of contamination that we need to know about? Microbial activity, besides just looking at roots, we have to look at the microbial side in order to sort of figure out that sort of overall below ground flux. So I have a couple of tools and techniques to look at how good the soil is at uptaking these root-borne carbon, which can then also be related to how much carbon is flowing through. So some soils might have more uptake and more competitiveness for that root-borne carbon. Both roots and soil roots tend to compete. And I also look at doing things like UAVs for precision agriculture and looking at both improving my monitoring by doing tandem above and below ground monitoring. So trying to really get at basically the differences or how the above and below ground connect together. And that's it basically for me. So this is my contact information. I am also going to plop it into the chat. So I love talking about roots. You want to talk to me roots more? I'll be happy to talk to you guys. Thanks.
Thank you, Cameron. So we will have a breakout room for our research partners. While we're waiting for that, I'll invite Dr. Meg McKay to take the floor. He's the director of GLIER at the University of Windsor and has been very busy chasing all sorts of samples. And I'd like to invite Mike to share screen. Yep, looks like it's up and running, Mike. So whenever you're ready.
Great. Thanks, Catherine. And I'd like to start off by congratulating you and your team with the Healthy Headwaters Lab. Great turnout tonight. And congratulations for developing this important network. It's really reinforcing, I think, the importance of our watersheds to the health of our Great Lakes. So making that important connection. So as Catherine suggested, getting back to what I really like doing, algal bloom research is refreshing. Having spent the better part of the last 6 or 7 months working on COVID-related research. So I'm going to share with you tonight just a little case study that we worked on last summer, along with my postdoc at the time, Tice Franken, and Ken Drouillard was part of this team as well. So I think most of you are probably aware the Thames River is located in southwestern Ontario, starts around London, runs through Chatham, and ends up in Lake St. Clair. It's a priority tributary under Annex 4 of the Great Lakes Water Quality Agreement related to the potential for phosphorus inputs into Lake St. Clair and Western Lake Erie. Let's see. Sorry, I'm just gonna. Okay. So September 23rd, 2019, we actually received a communication from the Lower Thames Valley Conservation Authority who reported, and I believe it was a Monday, so actually the bloom may have started over the weekend, but they showed up to work Monday. The waters had turned to green in downtown Chatham, and a team was put together fairly rapidly to respond to this, including LTVCA personnel, folks from the University of Windsor, and colleagues from the Ministry of Environment, Conservation and Parks. The bloom spread from roughly Thamesville, which is, I think, kilometer 64 of the river from the river mouth, down to around Prairie Siding, which is about 14 kilometers from the river mouth. So basically over a 50-kilometer stretch, but it was most intense over a 9-kilometer stretch in Chatham, from east to western Chatham. You can see these are just bottle samples taken at various stations along the way, and you can see the noticeable green color of the water, especially in those locations around the city of Chatham. So the bloom was composed mainly of this filamentous sort of sheath-forming cyanobacterium called the Phanoxaminum philosacui. There's a picture on the right-hand side. You can see we've poured some into a bucket. The way Phanoxaminum is often described, it looks like grass clippings if you're looking at it from the surface of the water. It is a potential toxin-producing alga. Luckily, during the bloom event in September 2019, we were not able to measure any cyanotoxins tested for microcystins, as well as antitoxins. But a Phanoxaminum can produce microcystins, it can produce antitoxins, it can also produce saxitoxins, which are potent neurotoxins as well. And the bloom actually had heterocyst, specialized cells in the filaments. These are cells which are suggestive of a nitrogen fixing bloom, but not always. And we actually found fairly high levels of nitrate in the water, so it doesn't reconcile well with having heterocysts, but we can discuss that in another moment. Okay, so here's the odd thing. We typically think of blooms occurring in lakes in our area. Katie Stamler showed a satellite image of blooms in Western Lake Erie. Also some blooms occurring in Lake St. Clair. But this is a river bloom. And really, it sort of goes against the standard line of thinking. So if we want to think about how blooms form, it's very simple. These are microscopic plants. They have very minimal requirements. They require sunlight. They require carbon dioxide. They require water, and they require nutrients. And the typical way of thinking about bloom formation is that blooms don't form in rivers, rather rivers serve as conduits for nutrient delivery into the receiving waters in the lake. And that's what typically happens in Western Lake Erie, especially with something like the Maumee River in Northwest Ohio. I spent most of my career working in Ohio, and so very familiar with the Maumee River watershed. We often refer to this area as the phosphorus superhighway. I use this because I look at this very similarly to the way I look at the Thames River watershed. The land use allocation is very similar. The watershed dominated by row crop agriculture, and the rivers serving as nutrient conduits, delivering nutrients into their respective receiving bodies. So the Maumee River, in this case, 220 kilometers extending from Fort Wayne, Indiana, to its mouth of Lake Erie in Toledo, Ohio. The yellow on the map is showing you land allocated to agricultural use, so probably in the 80% area for the Maumee River. And it's about a 17,000 square kilometer watershed. Now, we refer to it as the phosphorus superhighway because of this image, and this is showing you drainage in the Maumee watershed. 40,000 kilometers of tile drains, drainage ditches, etc., that eventually flow into the Maumee River and delivering nutrients into Western Lake Erie. We've seen algal blooms in the Maumee River as well, and just as we saw in the Thames River near Chatham last year. And what typically happens in these situations is the river becomes more like a lake. We have usually a period of relatively low discharge rates in the river. a period of very little precipitation. And in the case of the Thames River bloom last September, we saw a rapid increase in temperature, leading up to that bloom, which probably just provided conditions to allow the river to serve as a bioreactor. And so again, that's sort of the conditions we find typically if we see river blooms. When I've seen them in the Maumee River, we see them in the Thames. 2019 happened again this past summer in the Thames River. Same sort of bloom, one dominated by a Phantozomenon. And to finish up, Catherine mentioned that we have some breakout rooms, and she asked me to provide a spoiler alert. And so I'll be happy to talk about the COVID work we're doing if anyone's interested during the breakout. Thanks a lot.
Thank you, Mike. Yes, I highly encourage you to join our researchers in that breakout room if you want to hear more. There's plenty of, I have questions, so I'm sure all of you do. We now like to invite Dr. Ken Droulard to take the floor, as he's been also engaging in community-based research, and so he, he was a natural kind of person to reach out to invite to speak to this community here today. So, Dr. Droulard, you have the floor, and I can see your screen, so you're good to go. You just have to unmute.
There we go.
There you go.
All right, so I get to be the Monty Python, and now for something completely different, presentation in the room.
That's right, that's the one.
Very typically, I'm working with Catherine and I'm working with Mike. I also work quite a bit with Katie and others, including Justine, on water quality measures. But this is basically a sideline project that I'm doing that was a sort of a pivot for the COVID-19. And it is another community engagement project, so I think it fits in with the theme of this particular workshop tonight. And it basically has to deal with homemade masks and how we basically can come up with the best types of homemade masks. One might wonder how an environmental scientist who deals with water quality and bioaccumulation dynamics got caught into masks, and well, basically I got dragged into it, kicking and screaming by my spouse. So she is a quilter and a sewer, and she put together a volunteer Facebook group to produce homemade masks and donate them to healthcare workers right when the pandemic started. And her initial conversation with me was, how does our organization, first of all, select the best quality materials for our masks? What are the good designs? And you know, how do we test these masks to make sure that they're actually the best possible products that we can make and disperse free of charge? So we ended up getting some funding through the WeSpark Health Institute to carry on this project. And really, this is a 5-minute little primer, a little bit about our project and a little bit about the history of masks themselves. So I saw Dean wearing the very typical, surgical style disposable face masks that we typically see out there. These, the first published study was in 1897. And it was really, these things were designed to prevent surgeons from infecting their patients during surgeries. So really the intended use was to protect other people, not to protect the wearer of the mask themselves. And that was kind of the main basis for these surgical style masks. And of course, it became more prominent within the healthcare industry after 1927. This was aided quite a bit by the popularity of masks in the Spanish flu in the 1917 and 1918 areas. It wasn't until the 1970s that people started coming up with these things. This one is a Chinese version of the N95 respirator, but 3M came up with the materials that have these electric properties coupled with the filtration capacity. These are sort of the gold standards. They originally developed really for occupational health and in industry, people who were exposed to dusts and metal filings. They were basically designed again for a different setting than healthcare, and it really wasn't until the 1990s before N95s became a standard as a healthcare protection. oops. I have a little warning sign. Is my screen showing up okay, Catherine?
All I see is just the demystifying mask slide. So I wasn't sure if you were showing us a video or an image after that. I just see slide 2. So
Okay.
Just wanting to make sure that's what you wanted to show us.
That's fine.
And
Okay.
I don't see you as your screen. It's just,
You don't see my camera.
That's
Okay.
right. But I think that's changing now.
How
Slowly.
is that?
There you go. Yes.
So we didn't get to see our little prop.
That's right. I heard the
95.
propping. I heard some prop action. So I just wanted to.
All right.
All good.
Okay, so basically, N95s are the gold standards. We all know these are the best things going. They're able to filter 95% of particles, very small size particles, aerosol size particles, less than 1 micron in size. The surgical masks do much less efficiently. They're about somewhere between 50 to 60% of what these types of masks do. The problem is both of these materials were designed to be disposable. They're designed to be used once, throw it away. And of course, we're forced in supply interruptions to try and reuse these and try and decontaminate them. And these things were not really designed to be decontaminated. So there's a lot of research looking into that, but the results, apart from just waiting it out and using time as your decontamination, that seems to be the only real way that is possible right now. Now, when you go into the history of comparing these two, and all of the laboratory measurements that have been taking place, all of these laboratory measurements clearly show this is superior to the surgical mask. But a funny thing happened. In the late 1990s, a whole series of randomized clinical trials tried to basically demonstrate that in a real world setting with real healthcare workers wearing either this or this, that this would be much more superior as to what all of our lab measures had told us that they should. And it didn't turn out to be the truth. That was the weird thing. When we looked at healthcare workers that were either using surgical masks or N95 masks for the protection of influenza, and also in some major case studies where we had the SARS outbreak globally, they basically showed that either this or this both protected healthcare workers much better than not wearing a mask, but this one didn't do any better in practical performance than this one, which is a really strange thing because we're used to our measurements being, somewhat, proof of concept of whether something works. So that sort of takes us into the realm of homemade masks. And again, if we sort of contrast some of the properties and style, these are supposed to be disposable. They're supposed to use it once, throw it away. And of course, that leads to supply interruptions when we have a huge global demand for this PPE, as we already saw in the early phases of the pandemic, and we're starting to hear that during this second wave dynamics, that PPE shortages may be happening yet again, even in North America. So this basically takes us to the realm of homemade masks. Homemade masks are, again, not quite as high a quality. These are typically made of non-woven materials. They're designed to be water repellent because surgeries, they don't want to have blood spatter interacting with them. These have finer pore sizes compared to these. And these are basically, if we compare them in terms of filtration performance, the homemade masks are going to be a little bit less effective than these particular masks. On the other hand, we got a lot of really interesting attributes of these. We can wash these multiple times. Cotton, particularly cotton masks, are very robust to laundering. The hospitals use a wash cycle of 100 cycles before they end up reusing their medical grade cotton garments. So we know that these tend to be a little bit more sustainable. Another thing that's really interesting is where we started to compare actual quantitative fit testing in the performance, the real world performance, that we found that even though the theoretical filtration of this type of mask is better than your cotton fiber masks, in a fit test environment, they perform about the same. And that's really clearly what this is telling us, is how well that mask fits your face is just as important as what the materials it's made out of. So a little bit about our research project. I got pulled into this group that my wife had started up with a bunch of others. A bunch of Facebook groups basically collaborated together, and they joined together in what was now called the Windsor Essex Sewing Force, or WESF. And essentially they're donating, creating homemade masks, donating it to vulnerable groups. And to this stage, they've donated more than 30,000 masks. That's one for every 13 people in Essex County. And they've basically been donating these masks to the hospitals. They've been donating to some of the immigrant agricultural farm workers through a network. When the issue came up in earlier this summer, senior homes and residence buildings. And then they've been actually dispersing them to the city, who's been dispersing them to the homeless and low-income families, as well as Windsor Essex Health Unit. So basically, our role as part of this is to kind of engage in this three-tiered system of trying to develop best masks, best materials, and how to implement a quality control program within their volunteer framework, just to make sure that the masks that they're donating all meet up to specs. So one of the features that we've been doing is doing some characterization of different fabrics. I've sampled waters all over the world, and probably my most traumatic experience is spending 3 hours in fabric land with my wife, picking up 53 types of cotton fabrics. I'm still a little bit shell-shocked from that shopping experience, and I hope to never do it again. But we characterize all of these different fabrics, and with these electron microscope images, we're able to come up with pore sizes and gaps and kind of understand what their theoretical filtration capacity is. And we also worked with a industry partner called Second Chance CPR, where they were doing quantitative fit tests on their masks. And that's where we actually observed that, you know, our masks were performing about the same as what the standard surgical style mask was doing. We also ended up coming up with designs, and we generated an expert panel that consisted of basically infectious disease specialists, physicians, nurses, occupational health therapists. They all rated a set of prototype masks from different designs that are out there on the internet. Some of them were hospital sanctioned, some of them were produced by the members of WESF, and they all scored them, and that ended up creating two different, you know, the final prototypes that WESF now produces. The Essex pleated and the Windsor hybrid mask are the two designs that the panel has basically settled on.
I'm sorry, I'm going to have to cut you off if that's,
Yep.
Sorry, just in the interest of time. Really, it's just amazing information, and just really echoes a lot of what we've heard about local sort of solutions. I really want to make sure people connect with you in the meeting room if they have more questions, because there are opportunities for sure for people to engage. And we are coming up to our breakout room time. This would have been it, but we definitely want to make sure that our partner from Nature Conservancy Canada has the moment to share. Thank you so much, Dr. Droulard. And if I could just invite Karen Alexander from Nature Conservancy Canada to share our last lightning talk. And while she does that, Karen, if you want to go ahead and take the screen. After this, I'll kind of point you to the different breakout rooms. We've decided for people that, well, we've been able to set it up so people can choose the room that they are wanting to connect with. And then we will kind of, the advisory board and some of our lab will move to another room if we run out of time. We don't want to cut that conversation short. So in each of the meeting rooms, we have a Healthy Headwaters Lab facilitator who will be recording and facilitating the connections. And we might give our advisory board members just a few minutes in case you wanted to connect with the partnership groups before we meet again. So I'll come back in at the end after Karen's talk to just give you those logistics. Karen, are you ready to share, or are you?
Yes.
Yeah.
I thought I shared my screen.
I don't see anything yet, and I see your face now. That's good. And
Okay. Oh, there we go.
Yep, it started, and yep, the floor is yours.
Okay. Hi, everyone. My name's Karen Alexander. I work with the Nature Conservancy of Canada here in Essex County. So I'll just fly through some slides. I just want to introduce our program here in Essex. But first, in case there's some people on the line that don't know much about NCC, we are a national nonprofit conservation organization that has the mission really for a healthier future for all Canadians. And we work in conservation to achieve that goal and that mission. We're very focused, and we've been quite effective for over 50 years in our mission. And we do our work through these core activities on the screen. Conservation science planning involves when we start thinking about where our actions need to be placed on the landscape to ensure we conserve all of, or as much of, Canadian natural heritage as we can from coast to coast. Securement is where we're working on actually securing properties, land, either through fee simple. We also work with conservation easements to ensure long-term protection of Canadian natural heritage. And then we also, once we have a stake on the land or we own the property, we put a lot of effort into stewardship of the land. When NCC goes forward on a securement project, we fundraise an additional 20% of the value of that land purchased to put right into our endowment fund, which then funds stewardship work on those lands. So it does, it is a model that has worked and is continuing to grow for us and continuing to fund good active stewardship work on the ground. And then the final pillar, which is really important, is engagement and bringing the community into our work, but also finding ways for NCC to support and align to existing conservation work in the places where we are focused. So the science piece, I just wanted to show this slide to show everyone where we work in Canada. We use ecoregions to ensure that we are protecting as much of Canadian diversity, terrestrial biodiversity as we can. And of course, we are all sitting here in the Great Lakes region or ecoregion. We have 14 across the country, and the Ontario region natural area, this is a zoom in on the Great Lakes ecoregion, where we have 14 additional natural areas. So again, we're taking an additional conservation science planning step to zoom in on the places in the Great Lakes region that offer us the most opportunity to protect the greatest terrestrial diversity in this region. So we kind of break ourselves up into these natural areas and really focus our work in these places. And then, of course, down to the Essex Forests and Wetlands natural area, which is my position here at NCC, is implementing our natural area plan for this particular natural area. So we have 14 across the province, and each of these places has their own conservation plan. And this model really allows NCC to be quite flexible in how we apply conservation, how we address very unique threats that are occurring from one place of the province to another, from one place of the country to another. So we really are able to adapt, flex, and flex our programs to address very local issues. So here in Essex, some go through now a few of the things we work on in Essex, with the caveat that I will tell everyone, this year's been a fairly tough year for Essex region at NCC. I was seconded to Midwest region. So from June to December, Essex has gotten 1 day of my time a month. So things really did slow down, but we were able to do some good work, and we still have a very strong interest in moving ahead in the future. And that's going to be very focused on strategic protection and restoration. This is the most recent property we purchased in Essex in 2019, 102 acres in the headlands of Cedar Creek. So, you know, while we're looking at securing more land, we have some pretty lovely goals of several hundred acres of more land protected in Essex. We really want to be strategic about where we're doing that work and where we're transitioning land from current land use into conservation use over time. And this particular property was in a really great spot, right in the headlands of Cedar Creek in a Jetto clay area, which was already holding a lot of water and just naturally fit to slowly transition this back to wetlands, which was actually what the area was pre-settlement. So it's a pretty exciting property for us. We have 46 acres there that we will be restoring over time. And this is a picture of one wetland that had been restored by the previous landowner. So it's a project that also demonstrates that private landowner stewardship efforts on the landscape don't go unnoticed by the conservation community. We do have interest in protecting those efforts long-term as well. This year wasn't all for naught in Essex. We did get some work done. We managed 0.76 hectares of Phragmites at the new and Marion Girling Nature property, which was quite an effort that unfortunately I didn't have a part of, but I got pictures and it looked like a pretty fun day. And we did treat quite a lot of the Phragmites with herbicide. There's some left to do and we'll return next year. We pulled garlic mustard earlier this year. I did that by myself at the Oxley poison sumac swamp property that we own down in the southern part of Cedar Creek. We treated Canada thistle at both our Oxley property and the Marion Girling property, which I guess I forgot to show everyone where those are, but in the interest of time, I'll just keep going. I apologize. And then we also knocked out woody shrubs, invasive shrubs at the MGNR property. I think I just wanted to bring this up because it was a really great day for me. We pulled 150 kilograms of garbage out of the Oxley poison sumac swamp in February. And it was a really great day to get out there and kind of make that impact. We got our property sign up at the Marion Girling Nature Reserve, and we did some monitoring of both our properties. So we didn't do nothing, it's just it slowed down quite a bit this year. And then here in Essex, I think back to that, what I was mentioning about local threats and local issues. One of the big problems here in Essex is the massive infestation of Phragmites on the landscape. And this is an aggressive invasive plant from Europe that has taken a real strong foothold here across the landscape. So conservation partners that do work in wetland restoration or property securement are, we do the work, we get these habitats back, and then we're constantly fighting this plant to keep this plant out of those investments. So it's a part of our natural area conservation plan in partnership with ERCA to go ahead on this larger landscape level program for common reed. And it's really exciting for me because I have been helping with the Norfolk area, natural area, where NCC has been working very hard to eradicate Phragmites in the Long Point region. I think we topped 1,100 hectares of frag removed in those wetlands using a landscape level approach based in GIS. And this year I took that model and applied it on the Bruce, and it's getting up and running there as well. So I think with that internal expertise and the partners here on the landscape with the strong interest in addressing this issue and the impacts that we know are coming from climate change, we do want to get this plant addressed as soon as possible so our coastal wetlands and wetland habitats are as healthy as can be to manage the coming changes or to be resilient against them the best we can. So that's one big area that I'd like to see more momentum going forward in the near future. And then we also have our work and engagement. So here in Essex, that looks a lot like, well, what's on the screen here. We're working very closely with the Healthy Headwaters Lab to explore and support research to measure the impact of our work. And we're also looking for different opportunities to continue accelerating efforts, not only on our land, but other conservation lands and private lands. And then the Phragmites program is a great example of that, where we're not only managing on our lands, we're looking for funding, large funding to apply control implementation on other conservation lands, as well as private land control services to really sweep the land of that problem. So hopefully we can keep moving on partnerships and building that program here. And we do look forward to continuing engagement with First Nations and incorporating traditional knowledge. And then as COVID kind of hopefully goes into the background of our lives, we can begin engaging with the volunteer events on the ground. So NCC would normally have engaged volunteers to help with garlic mustard pulls, but this year we just could not. So we'll be looking in the future to start building those out again as well. So I feel like I went really fast, but I'm done, and thanks so much.
Thank you very much, Karen. Yeah, I will say that, all of the people that you've seen present today, we're working in different capacities. Thank you for sharing. I will just kind of point you towards, you should see breakout rooms being formed at the moment. And I will say that we have 3 of them. So